So today I'm going to talk about emerging zoonotic arboviruses, and I'm going to be focused on how they've come into the United States. I'm from Kansas State University, and I work for the Department of Diagnostic Medicine, Pathobiology, and the College of Vet Med, but my laboratories are all in the BRI because most of what I work with is BSL-3 agents. So as we all know, mosquitoes pose an extremely significant health threat to much of the population. And we work on quite a lot of different arboviruses. I've got a couple here that we work on listed. West Nile's been extremely important. It came in 1999 and it's caused about 45,000 cases. Chikungunya had 2 outbreaks. One caused about 1.8 million cases during the 2005-2007 outbreak in the Old World, and about 2.2 million cases since it's come into the Americas. And Zika has caused about 5,000 US cases since it came in 2015. Some other viruses that we work on are dengue and yellow fever. I'm not going to talk about them really today, but I am going to mention what we're doing recently with our Japanese encephalitis studies. Japanese encephalitis is not in the US, thank goodness, but we are checking to see what would happen if it actually did come over here, because it is a pretty bad disease. It has about 68,000 clinical cases and 20,000 deaths each year in its endemic areas. So just to kind of review, I'm sure most of you are familiar that the female mosquito is who has to bite in order to get a blood meal, and then she'll lay the eggs, which turn into larvae, and then into pupa, and then the pupae emerge into the adult mosquitoes. So this is a cross-section of a mosquito. Over at the top you see the head, and basically the female mosquito will feed through the proboscis, and if the animal that they're feeding upon has virus, the virus will go into the midgut, which is right here, and if the virus is able, it will go out of the midgut into the rest of the mosquito, which is called a disseminated infection, and then it can get into the salivary glands, and then it can be injected back into another host through the proboscis on a second feeding. So I'm going to talk about some factors that are related to how arboviruses emerge and reemerge, and I'm going to focus on the viruses I just mentioned and how they've come into the United States. So there's several factors such as travel, commerce, breakdown in public health measures, and then microbial adaptation is also important sometimes. So first I'm going to talk about West Nile virus. It's a flavivirus, and it was first isolated in Africa. And there's several other Japanese encephalitis serocomplex viruses, but I'll point out just Japanese encephalitis and St. Louis encephalitis here because I'll be speaking about them in a second. So whenever West Nile came into the US, it was in 1999, and at first it was thought to be St. Louis encephalitis because that was something that was circulating in the US already. It was later found to be West Nile virus. So West Nile virus causes a febrile illness, kind of very standard for these types of viruses. Fortunately, about 82% of the viruses cause asymptomatic infections. But of the ones that have a symptom or show symptoms, about 18% of them, there's a fatal case in about 4%. So it is actually a pretty serious virus to people who show symptoms. This is the total number of cases and the total number of deaths for the lifetime since West Nile has come into the States. So it starts in 1999 and ends in 2016. Over here you see the total number of cases. Total number of cases, and the scale is different. So the scale here is 0 to 12,000 on the case side, and it's 0 to 300 over here on the death side. But you can see how the death and the cases pretty much follow each other. So we have one peak here in 2002 and another one here in 2012 that I will discuss in a second. So the first peak is in 2002, 2003, and you can see that there's almost 10,000 cases just in the US. And the darker the scale, the more cases there are. And so there's over 50 cases whenever they're the dark. Basically, after 2003, everything kind of mellowed out, and it started to have about 1,000 cases per year. So this is 2009, there's 700 cases. 2010, there's 1,000 cases. 2011, there's back to 700. So there's kind of a lull in how much transmission is going on with the West Nile virus. But then there's a peak that I showed you earlier in 2012. So it's kind of interesting what possibly could have caused that peak. I don't think people really know what happened, but one of the factors could be a breakdown in public health because there was kind of a lull for 3 years before people decided, maybe we don't need as much spraying as we had. So anyway, in 2012, there was about 5,600 cases in the US, and they're concentrated a lot in the Dallas area and also Houston, Arizona, California. So this could be an example of breakdown in public health in the Dallas area especially. So 2016, it started to mellow out again at about 2,000 cases, and it's our 2013 to 2016, it's had about 2,000 cases a year, and it's kind of stopped at about 2,000. So maybe this is kind of the standard number of cases that we're going to have, and hopefully we don't have any more peaks anytime soon. So West Nile is different than the other viruses that I'm going to talk to you about because of its transmission cycle. It basically goes between a bird and the mosquito, and people are what they call dead-end hosts. They aren't really involved in the transmission cycle, and also horses are dead-end. So there are a lot of host species and birds, mosquitoes, animals that West Nile has been found in. So there's 330 birds, 65 mosquitoes, and there's a lot of different animals. Not all of them are involved in the cycle, but they have been found to have West Nile virus in them. So this is a lot different cycle than the other two viruses that have recently come into the US. Chikungunya virus came in next, and so I'll talk about it next. Basically, it was isolated in Africa as well. It's in a different family, though, than West Nile. It's in the family Togaviridae, but it causes a lot of the same symptoms. But one of the symptoms that's different is arthritis that can last for quite a long time. So this is a big problem with chikungunya. The transmission cycle, like I said, is totally different really than the West Nile cycle. It's involved with just the primates and the mosquito. So in the sylvatic cycle, it's monkeys, and in the urban cycle, it's people. So people and mosquitoes are really what's going to cause the transmission in the United States. So chikungunya is an interesting virus because it shows how international travel, international commerce, and microbial adaptation all kind of can come together to cause emerging and re-emerging of these arboviruses. So the first thing I'm going to talk about is the microbial adaptation. Basically, back in about 2003, I was looking at virus vector characterizing Aedes aegypti and chikungunya. So I was making infectious clones with GFP in them and just using them for basic mosquito virus vector studies. So what I did is I made the clones with GFP, and then I'd feed them artificially on these, the mosquitoes are down in these cartons, and this is just an artificial blood feeder. And what we would get is we'd be able to see chikungunya in the different tissues of the mosquito. So this is the midgut, salivary gland, eyes, more midguts. So it's just a good way to visualize the virus. Well, what happened during this time whenever I was looking at just basic science was an outbreak that happened in this blue line here from Africa into these islands in the Indian Ocean, into India, Indonesia, and all the way up into Italy. So this outbreak happened between about 2004, 2008. And one of the islands that was affected in the Indian Ocean was called La Reunion, and it's located here by Madagascar, and this is a larger version of it. And what happened was they noticed that the vector switched on this island. And so a group named from Schofenegger et al. Was following the genetics of the chikungunya as it progressed through the island of La Reunion during the 2005 outbreak. And what he noticed was this one amino acid change right here between an alanine to a valine on the E1 226 position. And they speculated that because there aren't Aedes aegypti on the island, but there are the Asian tiger mosquito, which is pictured here with the striped legs, they speculated that possibly this amino acid change caused this to preference the Asian tiger mosquito. So we had all of the clones, we had all the mosquitoes that we needed, so we were able to basically prove that this was correct. So this is a really good example of how the microbe can adapt and cause more disease in an area. So the Asian tiger mosquito on its own is a good example of how commerce plays a role in all of these diseases. Basically, this is a distribution map over time, starting in Asia, and then in the 1950s, the Asian tiger mosquito moved into a couple of different areas, but then during the 1980s through the 90s, it really expanded into the yellow areas, so we ended up with it here in the United States in the late 80s. And how we ended up with the Asian tiger mosquito was that we were importing tires. So this is a good example, again, of the international commerce. Basically, the tires had the larva in the tires, and they hatched out, and we ended up with the mosquito. So now in the United States, we have both the Asian tiger mosquito here, and we also have the Egypti, and both of these are chikungunya vectors. And if you look here, you can see that the Asian tiger mosquito actually goes a little bit further north, so this expands its reach into the United States for transmitting these diseases. Then in 2013, chikungunya left the old world and came into the new world, and it spread really quickly. December 2013, it went into this one island, and by the end of December, it was in quite a lot of islands. And in 2014, we had travel-associated cases in most of our 48 states, lower 48 states, and we had locally acquired here in Florida transmission of chikungunya. In 2015, we had, again, mostly travel-acquired or travel-associated cases, and then we had local transmission in Texas. For 2016, we haven't had any local transmission, which is good, but we're still getting a lot of travel-associated cases. So this just shows how viruses can move around just because of people. In Kansas, we had 2014, we had West Nile, and these were cases that were locally transmitted. But we had 13 cases of chikungunya brought in to the state by travelers. 2015, we had pretty much the same situation with West Nile and chikungunya. And then in 2016-17, we haven't had any chikungunya, but we've had 21 cases of travel-associated Zika virus. So we're still having about the same number of West Nile every year. So this segues into the last virus I'm going to briefly overview that has come into the United States recently is Zika. Again, it's a arbovirus. It's from Africa. It was discovered about the same time as the other ones in the mid-1900s, and it was found in Zika forest. So it's a flavivirus, which is similar to West Nile. So here you see West Nile, Japanese encephalitis, St. Louis encephalitis. This is Zika. So flaviviruses are all in the mosquito-borne section here. There's also tick-borne and no-known vector and insect-only flaviviruses. So Zika has a very similar transmission cycle to what you saw with the chikungunya. Again, it's mosquitoes and primates, both in the sylvatic and the urban cycle. So it's people and mosquitoes that are spreading it, again, in the US. So first, the virus was found in 47, and then the first human case was in 54. And up until 2007, Zika was kind of not really thought about much because it really didn't cause that much disease. It first really caused problems in 2007 on Yap Island. So it was found there. And then since then, it has moved into the Americas, where everybody knows that it's caused quite a lot of problems since 2015. So first it came into Brazil, but here's the United States. By 2016, we had 43 locally transmitted cases in the Florida area, but we had 3,000 travel-associated cases in all of the yellow here. So we are pretty well affected by this virus. Whenever Zika came into Miami, they started spraying, and it's good to know what mosquitoes you need to spray. So we decided to look at what mosquitoes are transmitting Zika. And we had a good idea that it was going to be Aedes mosquitoes because it was first isolated in an Aedes mosquito, and it's been found in quite a lot of Aedes mosquitoes. So it's pretty good guess that it'll be an Aedes mosquito. And this just shows, again, that it's probably Aedes. This is the old world, obviously, but everything in the blue here is a Zika virus vector, and all of them are Aedes, except for we have a little bit of Anopheles, Culex, and Mansonia, but everything else is an Aedes species. The dots just indicate that they're also vectors for other viruses, yellow fever, chikungunya, and dengue, but all the other ones are all Zika. So with that information, we decided that it's probably this mosquito that's going to be transmitting Zika. And we also had information from the 2007 in Africa that the Asian tiger mosquito can transmit it as well. So we wanted to look at Culex mosquitoes and see if they happen to carry Zika. We were doing this for a Japanese encephalitis project that I'll get to in a second. So basically we had the right virus, we had the right mosquitoes, so we were able to test Vero Beach mosquitoes that were collected for us in 2015, and we exposed them to Zika virus that was found from a human isolate in Puerto Rico. So it was the right virus, right mosquitoes, and they were not able to be infected, and we couldn't get pipiens infected either. So we're pretty sure that it's probably 80 species mosquitoes that are involved in this transmission cycle between the people and the humans here. So there's a lot of work that needs to be done. What animals are able to be infected. Richard Bowen's been looking at animals, and I'm not sure how many people have actually started looking at primates, but we need to do more work just to see what animals will be in these cycles. So if you remember, the chikungunya cycle is basically human and mosquitoes. The Zika is human and mosquitoes. West Nile is different because it's human, or they're the dead-end hosts, but the mosquitoes and the birds are really the cycle. Another virus that's very similar is Japanese encephalitis virus. And so we've been looking at the possibility of what would happen if Japanese encephalitis came into the United States. So we've been looking at, and the reason for this is because the amplifying host is here. We have the pigs, and we also have the birds. So the Japanese encephalitis cycle involves birds and pigs as the reservoir hosts, and the Culex mosquito, which is similar to the West Nile vector. It also has horse and human as dead-end hosts. So if Japanese encephalitis were to come in, it would be a very important virus because it has almost 100% mortality rate in the piglets, and the human cases would be very bad because 20 to 30% of the ones that show symptoms are fatal, and the survivors often have neurological damage. So JE is found in, as you would expect, Asia, and it's endemic in East and Northeast Asia. And in the 80s, it started to spread into India and then down south. And there has been a report that the genome of JEV was found in 2011 in Italy. So the question is, can it come over? Well, we have the right mosquitoes, and we do have the right birds, and Dr. Bowen from Colorado State showed us that we do have the right birds. He infected quite a lot of different birds with Japanese encephalitis. This is just one example where he found that the birds do have about a 4.5 log titer, and here this bird had about a 3 log titer. So that's enough for the bird to be involved in the transmission cycle. He also tested quite a lot of other ones, and he found that there's moderate viremia in quite a lot of other birds, and there's low viremia in chickens. He found no viremia in these crows and pheasants. So we do have the birds that are around, and we do have the mosquitoes. So we wanted to test our mosquitoes and see how competent they really were for Japanese encephalitis. So we guessed that it's probably Culex mosquitoes just based on previous epidemics and also laboratory studies that had been done. So we were looking at Culex pipiens and Culex quinquefasciatus, which is this mosquito right here. Culex pipiens is kind of northern US, and Culex quinquefasciatus is found in the south. And then we have this intermediate zone here in Kansas that is kind of a mix between the two. No studies like this had been done since 1946, and then we published our study in 2015. And basically all we did was we grew up our mosquitoes in our rearing facility. Then we infected them using artificial blood meals. And we ended up with our data. These are all going to look fairly similar. So we collected on 7 days and 14 days post-infection. We looked at infection rate, which is whole body titers, dissemination, whether it disseminated into the other organs, and then transmission, which is collecting saliva. And we were able to find that Culex quinquefasciatus at day 14 had about a 9% transmission rate based on the saliva. So this is just how we collect saliva. We have the mosquito, we remove the wings, legs, put the proboscis into oil, and we collect the saliva. And this is a picture of West Nile virus in a ball here in mosquito saliva. So we're assuming that this is kind of about what we would get with the Japanese encephalitis. But we're looking at it with titer and real-time PCR. But this is a pretty picture. Each one of these is an individual virion. It's a very good picture, actually. So what we did next was we looked at Culex pipiens, and we had 2 different pipiens. We had Michigan and California. And again, we looked at infection rate here at the top, day 7, day 14, and we looked at dissemination, day 7 and 14. And the only one that was different is at day 7, the pipiens from California seems to have a higher infection rate than the ones from Michigan, but this is not statistically different on day 14. But they are able to be infected and disseminate. We also looked at 2 other mosquitoes, Culex tarsalis and Culex restruens. They're able to be infected day 7, day 14, and have a disseminated infection on those days, but neither one is necessarily better than the other. So we also wanted to look at which genotype might be coming into the United States. There's 5 different genotypes of JEV. We have genotype 1, 2, 3, 4, and 5. And right now what's happening is genotype 3 is being displaced over time by genotype 1. So this graph is a little bit confusing. Up here is 100% of the genotypes. So in the 1930s, 40s, and 50s, 100% of the circulating JE was genotype 3. During the 60s, it started to be slightly displaced by genotype 1 here in the orange. And now in the 2000s, genotype 1 is actually the more prevalent genotype. So we wanted to look at these different genotypes. So we looked at genotype 1A and 1B and also 3. And again, infection rates on the top at day 7 and day 14, and dissemination rates on day 7 and 14. And this is in Culex quinquefasciatus. So the only statistically different thing that we saw was at day 7, the genotype 3 had a higher infection rate than the other 2 genotypes. So we also wanted to look at infectious dose, and basically this will just tell which strain will be more or less infectious. So this is the 50% line here, and basically each one of these is a different viral titer, so 4 logs of virus, 5 logs, 6 logs, etc. were fed to individual groups of mosquitoes, and then we just titrated them to see which ones could be more or less infectious. And you can see here that genotype 1A is, in fact, less infectious than genotype 1B or 3. So we also wanted to look at basically the same thing, but this time in the Culex pipiens. Again, we have the infection at the top, the dissemination at the bottom, and the 3 different genotypes, 1A, 1B, and 3. And for this particular experiment, there's really no statistical difference, but they are all able to be infected and disseminated. So the conclusion for the JE part of this is that basically we have these competent vectors, and they are present in North America. We have a lot of the different Culex mosquitoes, and there is a little bit of variation depending on location of how susceptible the different Culexes are. We saw that with the Michigan, California in the Culex pipiens. And infectivity of JEV strains belonging to the clade B genotype 1 and the genotype 3 seem to have a significantly higher infection rate than the clade A genotype 1. So there is a little bit of difference in the genotype, and genotype that may come into here. I mean, it could be either one, it could be 1 or 3 based on what's circulating currently. So my conclusion overall is that we can't control these factors. We have 3 great examples of how arboviruses can come into a new area and can cause a lot of human problems and animals. So basically the take home is I think we need to try to do a little bit of more proactive research and look at things that we think might come in, might cause problems, and things that can cause problems. So JEV is one of those viruses. There's a lot of other arboviruses that could potentially come in, but having some basic background on what people know about the viruses and how to kill the virus or kill the vectors of the viruses is important. So I want to thank my lab members. These are my current lab members listed here. Acknowledgements of all the collaborators. We have a lot of people that mail us mosquitoes from all over the United States. And we have a lot of people that help us with various parts of the projects. And then this is the current funding and questions.